For this meeting, I prepared an overview to talk about the chromatin accessibility analysis. This meeting is focused on the strategies and tools, as well as the common practice in this kind of analysis. So most of the information that I'm going to present is coming from three main papers. The first one is multi-omic profiling of the developing human cerebral cortex at single cell level. The second article is an integrated epigenomic and transcriptomic map of mouse and human A beta T cells development. And the third one is single nucleus chromatin accessibility and transcriptomic characterization of Alzheimer's disease. So just to make it digestive, I'm going to present indeed details of the first article, and then at the end, I'm just going to compare the strategies and tools used against the second and third article, so we can have, let's say, the big picture of what the strategy uses for these authors, as well as the tools and some differences that they apply on each one of the research. I will start talking a bit about the common vocabulary that is used in this kind of analysis. So the first concept is the states that we can see in the chromatin. The chromatin exists in two main states called euchromatic and heterochromatin. So we can see here the nucleus, and here is the chromatin that first is like present in a chromatin fiber. But then if this is extended, we can see here the two states, the euchromatic and the heterochromatic states. In the first one, it's relative open, as you can see here, some like region that it's open and is accessible for the transposase enzyme to measure this in absorbed fragments, and this is containing active transcript genes. While in this state, the chromatin is highly condensed, and so you can see here that as it is close, it's not as accessible as the other state. So typically, we can see here inactive genes. Also, it's a good opportunity to say that to study the chromatin can be studied with several approaches. So you can see here active marks that are usually mentioned in this kind of analysis. So here what we are seeing is histone modifications that are features also associated with active regulatory genes, but this kind of marks or epigenomic marks cannot be studied with the ATAC-seq protocol. So this is usually studied via different protocols like ChIP-seq. So this is one of the limitations. Also, ATAC-seq is based in an enzyme, a modified enzyme called TM5 or transposase mediated insertion. So we can see here in the cartoon that we have the chromatin, and we have here the enzyme with two indexes in each side. So it's going to bind the open chromatin regions, and these regions is going to be cut and is going to be amplified in sequence. So this is the information that at the end we have to build the libraries and to make the analysis. So let's say that the big picture or the overview of the methodology is to isolate the nuclei, the transposase TM5 to get the fragments. These are targeted fragments of the chromatin, so this is called PCA amplification, and to build the library preparation that then is take to the sequence, to next sequences, and then it's run a kind of analysis to measure some properties that are specific for this kind of assays. Another common vocabulary that we are going to find when we are running an analysis focused on epigenomics is that we have different elements of gene regulation. So basically, we have here a cartoon, and in this cartoon, you can see that this orange line or loop is the chromatin. So you have here a gene, the region of the gene, but close to the or nearby to the gene, we have promoters. There are usually two kinds of promoters. One are the core promoters that are nearby or almost close to the gene, and there are also another kind of promoters that are also close to the gene, but not as close as the core promoters. But there are also another kind of proteins interacting with the epigenomic state, when we have transcription factors and mediator proteins that helps to loop the chromatin. So this makes possible to the promoter region to interact with these distant control elements like enhancers, activators, or silencers. So one very common feature when we are studying epigenomics is that we are looking for promoters or some kind of enhancers. And the promoter region is one of the main quality measures that we can see in all these kind of studies. And this is because it's the transcription start size is close to the region of the transcription of the gene. And the promoter is let's say the pre-transcription state site. So here we have just a brief description. The promoter is located near to the transcription start size, contain a data box that it recruits RNA polymerase II to start the transcription. And then the insert is this kind of distant elements that make contact with the promoters when the chromatin is full, or it's like a loop. And we have also another elements that are called locus control regions. These are a longer range cis-regulatory elements that are called hoops that tends to control entire gene loci. So let's say that to provide a bit more details and understand what kind of elements are included in the analysis, usually these elements are classified in relation with the distance that they keep to the sum of the gene transcription. So for example, promoters are usually less than 2 kilobytes from the TST that we can see here. Enhancers are between 10 to 1,000 kbytes upstream to the 500 kilobytes, and the locus control region also is from tens of kilobytes to until 2 megabytes of distance. And this is just the big picture because recently there are other studies saying that likely there are enhancers that keep further distance, but it's difficult to infer because there are limitations in the ability of the enzymes to measure these kind of regions. So this is, let's say, in general, the roadmap of the typical ATAC-seq analysis. So we have pre-analysis steps when usually, for example, if you are using a multi data that contains transcriptomic and epigenomic data, indeed, the pipeline runs all these first steps that you can see here in the pre-analysis. So we don't have to do it by ourselves if we are using these kind of protocols pipelines like CellRanger-ARC, for example. But if you are running a manual analysis, when you are running an analysis with single-cell RNA-seq and single-cell ATAC-seq, and you manually are going to join the data, so you have to make the pre-alignment states, the pre-alignment QCs and further steps by yourself with using different pipelines. So after that you have the pre-analysis, that means that you have the count matrices to start the analysis. So usually the core analysis when we are working with epigenomic data is to locate peaks. The peaks are these regions where we have a lot of fragments, that are nearby to the region of transcription, or likely enhancers that are a bit further. But also we have from there an advanced analysis that include different kind of specific analysis that run from look for peak differentiation analysis, different differences between peaks, as well as the annotation of the peaks. I mean, it's like to transfer what cell types they are affecting, but also you can perform motif enrichment analysis or footprint analysis related with transcription factors. And well, if you are integrating this with other kind of assays like ChIP-seq or RNA-seq, well, you can build regulatory networks. So this is a cartoon just to emphasize that we have here the chromatin with the histones, and we have these open regions where is the TM5 binding the site. So we have here the nucleus on free fragment density, where it's going to be built theoretical like cure. So over this threshold, we can say that we have peaks, or we have located peaks that likely are regulating the gene transcription. And so also we can see here that likely we use this kind of diagrams to show that we have peaks associated with gene transcription region. And also we can identify if this corresponds to mononucleosome fragments in general. So this is a real example from the Habenula project when we can see here 3 tracks. For example, this is no habenula and lateral habenula. So we can see here that, for example, for no habenula, we cannot see peaks related with this gene associated with this gene region, while in the medial and lateral, we can see some kind of peaks. Also, there is reference that the width of the peak matters as well as the tall of the peak. And well, the other property that we can see in these studies is that close to the gene region, we can see here some like lines that are depicting the zone of the peaks. And well, one major step here is that we would like to see, for example, co-accessibility regions that are like regular and different gene regions. Now, after to present the common vocabulary that we are going to see, I'm going to start talking about these 3 articles. These 3 articles are the 2 first articles were making to make the developmental trajectories related with development and also some relation with some psychiatric diseases. But also we have different strategies. In some of them, they use multi-ome protocols, and other they use separate assays to make the analysis. And these articles are mostly focused, the first 2 in the dorsolateral prefrontal cortex, and one of these, the second one, with CHOP LB, is made in thymus from mouse and human. Okay, so as I said, I will start presenting more details of the first one, and then at the end, I'm going to compare the tools with the second and third article. So just to give you context, in the first article is just to present the multi-ome profiling of developing human cerebral context, single cell level. So this author, what they want to create is a multi-omic atlas of the human cerebral cortex across 6 developmental strategies. I'm not going to speak about the biology, but about of the tools used in this article. So what these authors do is that they use the 10x genomics single cell multi-ome protocol with ATAC and gene expression. They prepare 12 samples across 6 developmental stages, and they capture around 53,000 cells. After the QCS test, they have 46,000 cells. So they use a specific pipeline, the CellRanger-ARC version 1. Actually, we have version 2 something. And in this pipeline, what does is really prepare or do a lot of steps that I'm not going to explain in detail. But the point here is that the pipeline can take the gene expression library, align, count, and prepare account metrics. Also, the ATAC side of the pipeline align the fragments, call the peaks, and also prepares metrics with count of fragments. And we have a middle step that is called the joint cell calling, where both of these libraries are merged in one. So at the end, we have all the data that we need to run downstream analysis. And well, this is the pipeline that this author applied. So they, at the end, have this kind of report when we can see how many counts survived after to run this pipeline, as well as the quality of each one of the libraries. This is let's say, a very regular step in this pipeline. Now, what quality controls do they apply? Well, they use CellRanger and scATAC to run the analysis. So for the RNA side, they apply regular steps like filter cells with low gene counts, high mitochondrial gene expression, and other quality issues. And in ATAC-seq, they assess fragment size distribution. I'm going to talk a bit about it, as well as transcription, start size enrichment, and other standard QC metrics that are widely used during this analysis. So I'm going to focus in the quality controls applied to ATAC for these authors. So they apply what is called a nucleus signal less than 3, because, for example, in this cartoon, you can see that we have a nucleosome density But we have also a transcription start site that you can see here. And so we can measure with these kind of counts, or with the data that we have in the count metrics, we can identify the free region when the nucleosome is open. But also we can identify the first mononucleosome signal, the second the deconucleosome signal, and the third position of the nucleosome. After that, the enzymes start to degrade, and we cannot, let's say, accurately measure the next nucleosome positions. So when we have these analyses translated into, let's say, a normalized density of the fragments, we can clearly see that we are expecting to see in the nucleosome region around 100 base pairs. We can also identify the first mononucleosome signal and so on. So it's common to see that in many articles that authors are trying to keep the less than 3 nucleosome signals, the fragments that contains less than 3 nucleosome signal. Other metrics that are also used, and this is more, let's say, an arbitrary decision for each one of the authors, is to set a minimum threshold for the number of fragments that they want in each cell. So in this case, they apply number of fragments less than 1,000. And also they apply transcription start sites that we are talking about. But this is a measure that can be translated in an enrichment score. So I'm going to get back a bit of the vocabulary of concepts. We have the transcription start site. So the fragments can be like normalized, and we can set the transcription start site point in 0. And so the signal starts to decay when we are getting near far to the transcription start site region. So easily we can see that, for example, when this measure is normalized and set in an score and set in an enrichment score, we have this kind of measure that is running in the y-axis from 0 to A, that this is aligned with the encoded standard that says that we can expect to see this kind of TST enrichment score that is running from this measure. So it's common to see that at least we want an enrichment score that is bigger than 2. Because if you have enrichment score that is lower than 2, we really don't have, let's say, like a balanced pattern saying that we have at least one TST enrichment score related with the fragments. So this is a cartoon just to show that, for example, in the blue line, you can see the enrichment score that passed the threshold of 2, but the one that fails, that is less than 2, is really flat. So this is the scores that the authors apply. Also, they apply another metric that is fragment rate. I don't remember clearly the full name of this acronym, with an score of less than 0.2. But the thing with this particular metric, proportion of reads that fall within the cold peak region, is here. The thing with this particular metric is that actually it's not included in the CellRanger-ARC reports. So you have to calculate it manually. So it exists in previous versions, but now doesn't exist. Now, the peak calling, to make the peak calling, they use MAX2. Well, MAX2 was used to aggregate the single cell ATAC-seq data. They play a lot with optimized parameters like the model, like the chip, and also like the extension sites of let's say, of the open window to measure the peaks. And then to make the clustering, they use the Zebra-Denz-Signac approach using the weighted nearest network, but they use, let's say that it's not widely used. And I have not seen a lot of articles using the SEC transform, that is a kind of normalization that is trying to replace the previous, let's say, manual steps to normalize the data in the transcriptomic assays. And also they use the latent semantic index, that is the way to normalize the ATAC fragments, and this is a very regular measure in this kind of analysis. Also, let's say that to validate the the modality concordance between both assays, they use an adjusted run index just to see how well the data match. And one question that I had during this paper is why do the authors use MAC-S2 instead of scNAC, because we know that in scNAC has functions to do this, and they use scNAC in some steps. And so I found that likely, and this is my personal opinion, MAC-S2 is well-established algorithm, but particularly optimized for both ATAC-seq and chip-seq data. And in some forums, I found that this is especially helpful when we are trying to find consensus regulatory elements across cell types. In this case, they use it for developmental stages. So this is a particular property that is not, let's say, is not a strong point in scNAC to look for consensus regulatory elements that, let's say, to try to find if they are co-expressed or co-interacting. And also, there is another reason, possible reason, that this scNAC speed calling that is called by Colpix relies on MAC-S2 under the hook and with a more basic strategy, but it uses a lot of these properties in the way that are calling to pseudoball the peaks. Well, after that, there is very specific steps for the authors, like to run the pseudotime and look for the linkage between enhancer and genes. They use monoclonal tree to do the pseudotime inference. I'm not going to talk about cell trajectories, but they use it. And they also use Cicero, that is a tool to make correlation-based enhancer gene mapping. So I think what I can mention about this is that there are different ways, or let's say different approaches to call the peaks. So in this case, Cicero identify functional links based in correlated peaks. Let's say that we have a peak over here, another at distance. So it builds a matrix to measure this one. So let's say that a basic workflows is the Cicero uses as well the latent semantic index in the ATAC side, the same that uses scNAC, and builds a cell-by-cell distance map to model the co-accessibility, so it's able to generate co-accessibility networks to, let's say, identify putative enhancers in gene pairs. Okay. And the last steps that the authors run was the transcription factor motif analysis. They use ChromaTin. ChromaTin is used to quantify cell-to-cell variability in the transcription factors motif. And what I can say about this particular tool is that it's designed for both ATAC-seq, but also works well with multi, according with some authors, and it can also be well integrated using scNAC. So basically, ChromaTin identify active transcription factors, calculate transcription factor motif deviation scores, and infer the transcription factor activity patterns. Also, as a cell type, specific regulatory programs correlate motif accessibility with cell clusters of developmental stations, and also accounts for technical biases. So we have here, let's say, a profile of transcription factors. What kind of biases it's, let's say, modeling for this kind of tool, where basically the biases that they are trying to control is related with the PCR step, when the GC content bias have kind of preferences for this kind of content. So they make the algorithms has properties to correct this. Also, overall accessibility bias corrects the total accessibility peaks, because we can have more peaks related with one region and different kind of peaks related with another different region. So it models this. And also is mapping or sequence complexity bias, reducing the influence of low mapping regions. That is basically the same. And they use, to make this analysis, this specific analysis, they use the JASPAR 2020 motif database that contains a lot of transcription factors related with different species, organisms, and they then run the GWAS enrichment for map accessibility regions to SNPs for neuropsychiatric GWAS, and they also run the LDS regression, that is the heritability enrichment in ATAC peaks. So This is very specific for the study. So what we have here is that now, after to briefly explain the strategy and tools used, as well as to try to highlight what are common thresholds used to run the analysis, focusing attack, we have here the three articles. This is the article that I explained now. This is the second article in the second column, and the third one is the third article. So in the first article, they use a multi-ome strategy, and they use the pipeline CellRanger-ARC version 1, but the authors of the second and third article, they use separate strategies to count the metrics. For one side, they use CellRanger to get the counts for the transcriptomic side, and they use CellRanger-ATAC separately to prepare the metrics for the epigenomic data. As well, the third authors, they do the same. They prepare the data separately. It could be several reasons, because you can see here the versions that we are looking at. I think that it was not pretty well known, the strategy with multi-ome protocol also. And this can be explained in the way that they integrate the data that I'm going to mention now. For example, they use, in the first author, as I said, they use CellRanger, and they run the normalization with this new method introduced in the version 4 that is called SEC transform. While the second authors, to work with the transcriptomic data, they use the standard normalization because it was not introduced in this version of CellRanger. This method of transformation was introduced later in the version 4. So it's the same case with this is the same reason, but with the version 3.1.4. So here it was introduced, this methodology. Regarding the scATAC normalization, I think that there have not been like big changes. All the authors with different versions are using the same Latin semantic index. But what changed also is the way that the data are integrated. When we are using CellRanger-ARC, for example, it's very common to use the weighted network. They use particularly also, let's say, it's not new, but it's not pretty well, how do you say, not used for many people, the smart local movement. These are the kind of methodology applied to this weighted network. While the authors, for example, for the second article, the data integration that they do was really manual. They use monoclonal 3, they use MAC-S2, and they use Homer. While the third article, they use specifically almost the same tools. They use monoclonal 3, MAC-S2, but with C0 for the motif enrichment, together with chromatin. Chromatin was also used, as you can see here, with the first author. So I can say that the strategies used for these authors are not just, let's say, seems that were chosen regarding the kind of methods that exist at that moment, because that have been in every step that is released a new version, we have new methods available. So they are likely picking up the methods that are well established at that moment. And also, well, after to have CellRanger-ARC, where the benefit of this specific protocol is that we are not making inferences, because we have barcoded data from both sides of the modality, so we have the same information for the same cell in both modalities, transcriptomic and epigenomic, while here, what is done is kind of inference that is going to be translated from the RNA to the attack. So this is also, it is also a common strategy used in this kind of analysis. So some nuances to consider here is how much abundance is needed to detect cis-regulatory elements. This is a very common question. Well, likely we need, according with this article, we need around 1,000 nuclei to identify over 80% of these accessibility regions in each cell type. And this is consistent with a lot of studies. Also, another question that people is asking a lot in the forums is if are cell barcodes accurately called in the multimodality protocol, like CellRanger-ARC. Well, what I can say here is there is a very relevant step in the algorithm. That is the cell calling algorithm. Is the name of the step that uses paired data to distinguish cells from non-self background. But if the barcode fails in any of the filtering steps, so the data is basically masked from the local total set of barcodes prior to the cell calling. So what this means is that you are going to sell a lot of cells, because basically these data are masked if they don't pass the thresholds. So you have alternatives, but this is what it is. And the third point is that our chromatin accessibility peaks proximal to the TST. I mentioned that not necessary, but also I want to add that only 22% of the peak genes links occurs between an ATAC-seq peak and the next genes. So it means that many of the regulatory elements are not as close as we think. And also, for example, this author, Keiji Su, they say that he have found that mostly many of the regulatory elements skip at least one gene and then linearly continue to the other gene. So this is some things that we have to think about when we are running this kind of analysis. And I'm going to finish with this slide saying that, well, there are other tools, of course, also based in Python, like scATAC too. But there are another one that I think is the SEC multi-pack. It's another tool that is wrapping the information from both modalities, but there is also this another one that is called ATAC graph that also can model this kind of data. And in summary, single-cell ATAC-seq is going to enable us to discover regulatory landscapes by profiling the chromatin accessibility at single-cell resolution. Multi-omic approaches integrate the chromatin accessibility, but also the gene expression from the same nucleus. This is a big benefit. Enhancing the biological resolution, while separate analyses offer greater flexibility, but you need a lot of work to do in this kind of strategy. The tools founded in this analysis are CellRanger, scATAC, MAC-S2, cis-0, and Chromatin, seems to provide a robust framework for data processing, for clustering, for enhancer gene linkage, and transcription factor motif analysis. And well, these case studies across brain, and in the case of the thymus, that is based in the immune development, highlight there are conserved regulatory logic, cell type specific cis-elements, and disease associated regulatory variants associated with this kind of analysis. The field is rapidly evolving, so we need to keep exploring tools, interactive tools, functional assays that can influence the way when we are looking for this kind of information. And that's it. Thank you. Do you have some questions?
Thanks, Cynthia.